Hi, I'm Abby Nimick. I'm in your Monday, Wednesday, Friday 1 to 150 bio lecture, and we're going to talk about the structures of proteins. So in this example here, this is the primary structure. So the definition is a linear sequence of amino acids. There's only one type of bond in this structure, and it's a peptide bond that is formed between amino acids, the amino group and the acetic carboxyl group. The amino acid, parts of the amino acid that are involved are the amino acid group and the acetic carboxyl group. So, yeah. Peptide bonds have a similar strength to double bonds due to the attraction to nitrogen and the carboxyl group, so the nitrogen and the carbon. Overall strength, it's pretty weak. So then this is the secondary structure. The definition is the arrangement of polypeptides into a regular alpha helix beta structure or random coil configuration by the formation of intramolecular hydrogen bonds along with the length of the chain. So pretty much they're like beta and alpha plated sheets, so they're like folded. This is due to the hydrogen bonds found inside of them along with the peptide bonds. So the peptide bonds are found in primary structure and they hold together the amino acids, and the hydrogen bonds create the fold, the pleated sheets. One example of this is myoglobin, which is a protein that carries oxygen to muscle cells. So parts of the amino acid that are involved are the carbonyl and amino acid groups of the backbone, where the hydrogen bonds are formed. So here. The strength, the peptide bonds are the same as in the primary structure, but the hydrogen bonds are not very strong typically, but in this case they hold together the secondary structure. So overall they're stronger than the primary, but still not the strongest. This is the tertiary structure. The definition is tertiary structure is the three-dimensional shape of a protein, so it has multiple amino acids, like chains, that make up the tertiary structure. So the types of bonds are peptide, hydrogen, and ionic bonds. Peptide and hydrogen bonds are found in the secondary structure, as I said before. But the ionic bonds create disulfide bridges, which make the tertiary structure different than the secondary structure. Disulfide bridges connect sulfur-containing side chains to cysteines. And then parts of the amino acid that are involved are the sulfur-containing side chains. The strength, bonds are very strong due to the disulfide bridges. So overall, they're very strong. And then the quaternary strength is right here, and it's made up of multiple tertiary structure proteins. So the definition is an arrangement of multiple folded or coiled proteins that form a multi-subunit complex. And like I said, it's consisted of multiple polypeptide chains. The bonds are peptide bonds, hydrogen bonds, and ionic bonds, which are all found in the tertiary structure. The difference between tertiary and quaternary structure is the hydrophobic bonds at the core of the quaternary structure, which are away from water because they repel water, and they stabilize the folded state of the structure. The R group of the amino acid is involved in the quaternary structure, and overall the structure is very weak and it's prone to denaturing. So if you add heat or change the pH, the bonds will break and it'll denature. And those are the 4 types of protein structures.